So this is a disclaimer. We have been collaborating in the development of JAK inhibitors with Pfizer for many, many years. And the NIH holds a patent on targeting JAKs, not the development of the drug, but JAK as a target. This is an introductory slide, which probably is going to be redundant for this audience, but because all of you, and that's why you're here, know about the importance of cytokines and cytokine receptors in the development of autoimmune diseases. And they're so important that the most successful drugs that have been developed in the past few years have actually been targeting these receptors. All these biologics that we are going to be here in the next few days. The question that for us working in the immunology field, but looking more at the signaling point of view, that's what I've been working for the past 20 years or so, it was, can we actually target the signaling that it's downstream of these receptors? And the problem is that, like Luke was saying before, when you start talking about signaling, it's almost like talking about metabolism. People go nuts because they are seeing something like this. It's complex. They don't understand it. There's a bunch of molecules here, which the acronyms drive me crazy. So I don't want. It's a black box. I just don't want to deal with it. There are, in fact, difficulties because we're dealing with molecules with different effects. Some of them are enzymatic. Some of them are not. Some of them could be just simple adapters. They kind of, works as docking stations. So yes, it is difficult to target signaling molecules and signaling in general, but it's not impossible, and I hope I'll be able to convince you during my presentation. So in terms of immune cell response, there is one particular event which is actually very, very important for most of the immune cells in terms of signaling and enzymatic event, and this event is phosphorylation. It's critical, especially at the proximal events, but also even downstreams in different pathways, for receptors on the surface of B cells, T cells, and so on. So phosphorylation is the subject of our research, has been the subject for our research for the past few years. And it's actually, like metabolism, is actually very, very important because studies of phosphorylation, so the attachment through the utilization of ATP of a phosphate group to an hydroxyl group was actually in a certain way featured in the award of 4 Nobel Prizes. So, not a small piece of science here. So phosphorylation acts essentially in 2 ways. There's an action of a kinase and a phosphatase. The kinase normally attaches the phosphate to the hydroxy group and the phosphate removal. And it can actually go in both ways for the activation or deactivation of a molecule. So the kinase can phosphorylate the residues and turning on the subsequent substrate, or vice versa. You can actually turn that molecule off by phosphorylating the substrate. So it's interesting in no matter which way you look at it. And then you have, with the advent of the genome era, we were finally able to realize how many kinases we have in our body, and we actually have 518 protein kinases. So this is the tree of the kinome. It's probably even more complicated than you look at the picture that I showed you before about signaling, and it scares even more so. They're divided in 8 major groups. There are about 90 tyrosine kinases, and I'm highlighting the tyrosine kinase because I'm going to focus mostly on tyrosine kinase during my presentation. These kinases are all, of course, critical for signal transduction and the signaling events. And since you already know that I'm going to be talking about JAK kinase, in these 518, 4 are JAKs. So the question that we had 15, 20 years ago when we were starting working on the development of possible inhibitors of JAKs is it possible to generate pharmacologically useful inhibitors of tyrosine kinase? Most of the kinases actually bind ATP as have an ATP binding site. So is it possible to develop drugs that blocks ATP? And this is even more complicated because the kinase structures is very conserved among all of the kinases that I talked to you about. And kinases are not the only enzyme that utilizes ATP. there's a bunch of other molecules that utilize ATP as a substrate. So even more complicated. Well, it is possible, and that's otherwise I wouldn't be here, of course. The poster child, the molecule that actually started everything, of course, is imatinib, the very strong precedent for all the studies they're going to talk to you about, developed as a competitive antagonist of ATP to block the ABL kinase, and so it's widely used in the treatment of leukemias. But there's another, besides ATP, imatinib, a large number of molecules followed the success of imatinib. Most of them developed to treat malignancies. And in the past year and a half, 2 more have entered the clinical arena. And these 2 molecules that I have highlighted in red down here at the bottom of the slides are actually targeting the JAK kinase that I mentioned before. So going back to the cytokines and the signaling pathway, the so scary signaling pathway. Cytokines signal utilizing kinases, either tyrosine kinase like the JAKs or other kinases like IKK, GS3K, and so on. Ultimately, all these results in activation of transcription factor and gene induction, and therefore there is a regulation of cell behavior. But everything is controlled up here, especially in the type 1, type 2 cytokine receptor. The JAKs are very, very proximal. They bind the receptors, and they actually control essentially the downstream signaling pathway. To go a little bit more into the JAKs, for the people that are not familiar with this family of kinases, there are 4, as I mentioned it before: JAK1, JAK2, JAK3, and TYK2, just to complicate your life a little bit more. The basic knowledge is these molecules work in pair, either attaching to the receptor chains. They could be either the same JAKs attaching to, for example, the IL-6 family of receptors here, or different JAKs, but they actually bind to the receptor and they phosphorylate the receptor themselves and the subsequent substrate. So how critical are these molecules? They are very critical. Again, otherwise I would not be here. But this is actually a paper that essentially started the whole field, or one of the papers that started the whole field, because what this paper showed back in the mid-90s is that when you have mutation in the tyrosine kinase JAK3, you essentially phenocopy the same clinical features of patients that have mutation in the gamma C of the IL-2, IL-4, 15, and so on, the common gamma chain of the IL-2 receptor, and these patients have severe combined immunodeficiency. Same thing. So JAK3 or the gamma C mutation results in a very similar disease. And in that paper, this sentence was put there, I think almost as a filler. We always like to conclude papers giving the hope for development of drugs, but saying that, this current study suggests that agents that can activate JAK3 can be potent immunosuppressants. Well, it turned out to be probably a very prophetic sentence. Fast forward about 19 years. As I said, we have now 2 JAK inhibitors that have been approved by the FDA in the case of Tofacitinib in Japan, but not in Europe, as was just mentioned, for the treatment of malignancies in the case of ruxolitinib, in particular myelofibrosis, polycythemia vera, and in the case of Tofacitinib, so far the approval has come for RA. There are other in the footsteps of these 2 compounds, baricitinib and pacritinib and lerstranitib, following again, targeting again JAKs, in a more or less specific way, while I'm going to get into the details in the next few slides. Recapitulating about the targeting of the JAKs, the JAK inhibitors block the ATP binding. The receptor per se don't transmit the signals. They need this kinase in order to transmit the signals, and that's where the inhibitors come in. They block the ATP. So how can this drug be specific, and how the selectivity or specificity of this drug is important, and how important is the selectivity among the JAKs, and is there a kind of selectivity? So that's what I'm going to address in the next few slides. In terms of selectivity of the JAK inhibitors, and this slide that refers to a paper that came out about 8 years ago on Tofacitinib. As you can see, this is an in vitro assay done with the crystallized kinase domain. It's not the full JAKs that are here. They're looking at here. There's no crystal structure so far of any of the JAKs that have been resolved. But this in vitro assay has been validated, and I'll show you some data later on about that. And so these slides show that Tofacitinib is actually relatively selective for the JAKs. It inhibits JAK3 and JAK1. JAK1 is not listed here, but the IC50 for JAK1 is very, very similar for the one that is listed for JAK3, and it has very minimal effect on other kinases. What does it mean? How minimal is minimal? Well, this is a comparison of some of the kinase inhibitors that I showed you in one of the slides before, where I listed all the FDA approved. So imatinib is quite specific. Other that are followed imatinib are less specific, and just to give you here as an idea, this is used to be our favorite kinase inhibitors working in the lab about 15 years ago. Storosporin is actually very non-specific, but this is just to give an idea. These are not only FDA approved, but actually they are used in the treatment of malignancies. And actually, the fact that they are not so specific, it actually turned out to be an advantage, because we can treat patients that become resistant to imatinib with less specific drugs and actually still have responses in case of some malignancy. So overall, what I showed you before in the previous slides about Tofacitinib and the JAKs, it shows a good profile in terms of specificity towards the kinome. So summarizing a little bit about what we know so far about the mechanism of action of these JAK inhibitors, more or less the one that we're working on right now, have been developed and studied so far, are very, very similar in the mechanism of action. They block different JAKs. They have major effects on T and B cells. Their action results in impaired differentiation of Th1, Th17, and Th2 cells. There's no decrease on overall CD4 T cell population, neither seen in the animals, in the patients. There's been an observed decrease in the number of circulating NK cells. They do have a metabolic effect. Not only, I put this in not only because Luke is here, but also because I'm going to show you some data about it. What is interesting, because the fact that these JAKs block, although with different IC, different kind of JAKs. They not only block, like in the case of Tofacitinib, for example, the adaptive immune response, but they also block the innate immune response. The activity of IL-6, interferon gamma, and type 1 interferons is blocked. As a result, they also block the production of pro-inflammatory cytokines. So they work on both arms of the innate response. So you heard about before from Luke about the Warburg effect, and we actually looked at the effect of JAK inhibitors on the T cells, because the Warburg effect not only was studied in tumors, and as Luke had shown you in innate cells like macrophages, but it was also studied in T cells. And so naive T cells essentially mostly utilize oxidative phosphorylation that you heard about before. So the glucose, it's metabolized through the mitochondria to produce ATP. And activated T cells instead prefer to use glycolysis. There is a production of lactate. There's no longer uses of the mitochondria, and both fatty acid, glucose, and amino acids are utilized to generate the ATP. Okay, that's essentially what I just told you. And the glycolysis-dependent energy production actually is maintained and pushed forward by the presence of IL-2. and that prompted us to ask the question, does Tofacitinib affect the T cell metabolism? And we used a similar system, actually the same system that Luke talked to you before, this machine called a seahorse, which allows us to measure the oxygen consumption rate, essentially the oxidative phosphorylation, or the extracellular acidification rate. So essentially we go and look at lactate. And so to distinguish between the OXPHOS and the glycolysis. And so by using specific inhibitors of this transporter, we can actually look at the basal respiration of a T cell, and then eventually look at the maximal respiratory capacity. So by looking at the oxygen consumption, so at OXPHOS, and by taking advantage of drugs like oligomycin and 2DG that Luke had talked about, we can actually look at glycolysis and therefore look at the extracellular acidification rate. So just to show you a couple of pieces of data about our most recent and unpublished studies, we'll look at the effect. This is the system we work with, taking the T cells, purify them, stimulate 3 days in vitro with CD328, and then come in with IL-2 plus and minus Tofacitinib and look at the effect of Tofa in this event. We actually also look at the first part, and I'm not going to show you the data, but we have data showing that essentially Tofa does not affect in any way the metabolic effect driven by TCR. But just to summarize, these are the data. We see when you have the T cells are in the presence of IL-2, the basal respiration is increased, and the maximal respiration is increased. If you add an anti-IL-2 antibody in the culture, essentially when you start the culture right here, you essentially bring down both the basal and the maximum respiratory capacity. And Tofacitinib essentially phenocopies what we see with anti-IL-2, decreasing both basal and the maximal respiration. What was interesting is that when we look at the glycolysis, we found pretty much the same result. The anti-IL-2 and Tofacitinib behave very, very similar. We were actually able to decrease maximal glycolysis by treating cells with Tofacitinib. We then did RNA-seq and look at the gene expression changes both for oxidative phosphorylation genes involved in the TCA cycle, as well as genes involved in the glycolysis pathway. And as you can see, cells treated with Tofa essentially decreasing the expression of some of the key genes that are activated in both the pathway. So just to summarize, Tofacitinib does not inhibit TCR-dependent mitochondrial respiration, but it does inhibit both oxidative phosphorylation and glycolysis in response to IL-2. The OXPHOS and glycolysis genes are also downregulated by Tofacitinib. So overall, there is an inhibition of T cell metabolism, which is an aspect of a mechanism of action of Tofacitinib, which is quite surprising because we had no idea that this was the case, although JAKs being, as I said, the very first step that are involved in the activation of a T cells in response to a cytokine. This was totally unknown to us. Moving forward to the JAKs, so what's the future? It's also difficult to predict what's the future of a class of drugs. What's happening right now is that companies are developing more specific inhibitors, inhibitors that can only target one JAK at a time. Hopefully, this will allow us to do more mechanistic studies, not only in the case of metabolism, but also try to figure out what JAKs are more important for one specific disease or one specific pathway. This actually also brings about the question about how should we think about this class in drugs of maximal safety and so on. Are these like biologics? Will replace the biologics? Will be replacing steroids? Can they be used as steroid-sparing drugs? Can they be used with different modalities? And in fact, there are a couple of studies already ongoing, both for ruxolitinib and tofacitinib as topical agent to replace steroids in the case of psoriasis, for example. In terms of selectivity, we have no idea what's the future of more selective JAK inhibitors will bring to us. Possibly there could be useful for specific disease more so than the non-specific, but again, we don't know. Or maybe can be used in different phases of the disease as well. And as you can see, there's a bunch of there are in the pipeline. I'm just listed here, but there's more and more coming. A few notes about the toxicity of this class of drugs. Clinical studies have been performed. Infections were observed, very few opportunity infection, a little bit of an increase in herpes infection. Cytopenias and anemia, as well as leukopenia, have been observed, presumably related to the block of JAK2, and this has been observed more so with ruxolitinib because it's a much more specific JAK2 inhibitors. We don't know anything about tumors, possible increase in tumor numbers, and this is actually one thing that it's really worries when the NK decrease was observed. There is an increase in lipids, very similar to what has been observed with tocilizumab. The mechanism, it's absolutely unknown. There's anything that has to do with the blockage of JAKs, we don't know. As well, an increase in creatine transaminases. But again, we still don't know if this is related or unrelated to JAKs. Since the title talks about autoinflammation, I'm going to put I felt compelled to present you a few slides about the use of these inhibitors also to treat other diseases. And this is an autoinflammatory diseases that has been studied at NIAMS by Rafaela Gold-Batmansky, a colleague of mine. And this was reported about a year and a half ago. She reported this disease called CANDLE, which stands for chronic atypical neutrophilic dermatosis with lipodystrophy and elevated temperature. It's the results of a mutation on immunoproteasome molecules, PSMB8, PSMB4. These immunoproteasomes are deputized, although they are known as immunoproteasome, to deputize the degradation of molecules and the presentation of peptides MHC, but the reality is this very little is known about it. What was observed in this patient from the very beginning was the presence of so-called interferon signatures, as you can see from this heat map, in which there are 9 CANDLE patients reported, and we have few controls next to it. There is an increase in these sets of genes here, which are it's too small and too difficult to see for you and for me as well. But you have to believe me, these are interferon-related genes. And so because of this and because of some of the data that I'm going to show in the next slide, a trial was started utilizing baricitinib in these patients. And I'm going to show you one piece of information about that as well. One of the bases that prompted Rafaela to start the trial with JAK inhibitors is depicted here. This is a healthy control and looking at phosphorylation of STAT1. And as you can see here, the basal phosphorylation for a control and the basal phosphorylation of STAT1 for a CANDLE patient, there is a shift toward the right for the basal phosphorylation in the CANDLE. Not only that, when you stimulate with interferon gamma, the cells seems to be hyper-responsive. There is a clear increase in phosphorylation of STAT1 response to interferon gamma in the CANDLE patient. This response can be controlled by, this is of course an in vitro assay, by treating tofacitinib was used in this case, and so we can bring down the level of STAT1 phosphorylation by treating with tofacitinib. So as I said, Rafaela started the trial a few months ago, and these are just 3 of the patients that have been treated with increasing dose of baricitinib. And what you're looking here is the expression of some of the interferon signature genes that were in the previous slide. And as you can see, there is a clear decrease of these genes in response to the JAK inhibitor. So these drugs can actually be used not only in autoimmune diseases, but have the potential to be used in other pathologies, which could be mediated by interference, in this case by other cytokines or molecules that utilize the type 1, type 2 cytokine receptors. So what about other kinase inhibitors? There are, again, 518 kinase, so plenty molecules to look at. I'm going to show you a few data about Fasenmyer. You're going to hear about that probably later on during this meeting. But molecules have been developed not only to target the spleen tyrosine kinase, but also in the Bruton tyrosine kinase and other kinase that are utilizing in signaling events in immune cells, the MAP kinase, and I also have a slide about that as well. But RAF has been very successfully targeted for melanoma, and actually there are trials ongoing for possibility of target RAF in other diseases as well. About Fasenmyer, the spleen tyrosine kinase is actually located in a cartoon that I have here up at again, one is the proximal event. It's utilized by many immunoreceptors in B cells, neutrophils, mast cells, and therefore it was right from the beginning a very good target. And in fact, the drug Fasenmyer was studied, and in phase 2 RA, the ACR response was fairly good. And again, I'm not going to go into the detail. You very likely going to hear more about that later today. But both the ACR 20, 50, and 70, and DAS28 show very good efficacy for these drugs. Some of the adverse events were diarrhea, hypertension, neutropenia. Again, not surprisingly, considering the importance of these kinase for neutrophils. No opportunistic infection. And there are ongoing trials for these drugs in other diseases, mostly malignancies and cancer. And also there a report a few years ago about the utilization of Fasenmyer along with for patients that have failed treatment with biologics. So other molecules I already mentioned, looking at the B cell receptor, targeting the B cell receptor, but we're not stopping just right here. In general, we also you have to consider when you're targeting signaling, we actually, in fact, already have molecules that are very efficacious in targeting signaling. We have calcineurin inhibitors like cyclosporin A, and we have molecules that target TOR, and again, these are molecules that potentially have a metabolic effect, very likely by targeting target rapamycin. Here we are actually working not only blocking signaling events and gene expression, but also metabolism as well. A few notes about targeting MAP kinases. As I said, frequently the BRAF is mutated in melanoma, and that's why the molecules have been developed. And there are first as well as second generation BRAF inhibitors that are being developed. Both T-cell receptor and IL-2 activate RAF, so there is a potential interest also for immunologists in these molecules. The utility of these drugs in rheumatological disease, it's unknown. And I don't have to talk to you much about P38 MAP kinases. They have been studied and for a few years now. Several clinical trials have been completed in RA and other autoimmune diseases. The clinical efficacy for these drugs has not been shown to be very effective, unfortunately. So Other molecules are now being targeted maybe a little bit more upstream. MKK6 and MKK3 have become probably have shifted the interest of people working in this, looking at this pathway as potential targets. Just to summarize, essentially, we have a bunch of kinase inhibitors that are in the clinics already. Imatinib being the first one, but other following its footsteps. And in the case of autoimmune diseases, now we have Tofacitinib and ruxolitinib as JAK inhibitor. Other that are coming along, Fostamatinib, as well as others that I showed you before that can also be used not only in autoimmune diseases, but also in autoinflammatory diseases as well. And I would like to conclude with one slide which has nothing to do with the kinome, but the JAKs have as a downstream target the STATs, and there are a bunch of STAT inhibitors that are up and coming and have been actively studied. Some of them are listed here. Some of them are drugs that are well known and utilized, but they are kind of being repurposed as STAT inhibitor. And as you can see, the most commonly targeted STAT is actually STAT3 because of the role of STAT3 in many malignancies. But STAT3 is a key molecule, as you just heard, for the signaling of, for example, IL-6 and IL-21. So there is a potential interest in targeting STAT3 also for autoimmune diseases and not only for cancer. And this was my last slide. I'll just stop here. There's a few people that I have to acknowledge that have done some of the data that I show you. Yasko working with me on the metabolism and the effect of Tofacitinib of metabolism of T cells, and Adriana and Yin, which are responsible for the studies that I show you on Candle. And again, we have been collaborating with Pfizer for quite a few years. So I think I'll stop here and I'll take any questions.